Okay, so this is the electron transport chain mini lecture. So remember that during glycolysis and the Krebs cycle, we made all those electron carriers, the NADH and the FADH2. And those electron carriers are carrying high energy electrons from the breakdown of glucose. During the electron transport chain, those high energy electrons are used to transport hydrogen ions across the inner membrane of the mitochondria. And the hydrogen ions are moved from an area of low concentration to an area of high concentration. So that's active transport. If you remember from our plasma membrane unit, active transport requires energy because you're going against the concentration gradient. So the high energy electrons are the source of that energy. This is just a review from the cell organelle unit, but a mitochondrion is structure is a highly folded inner membrane surrounded by an outer membrane. And that inner membrane has a matrix, and there are intermembrane spaces created by that folding. So really for the electron transport chain, what we're looking at is this highly folded inner membrane this highly folded inner membrane here, and this matrix, and this intermembrane space. Okay, and here we are again. So we've got this intermembrane space, the actual inner membrane, and now it looks a lot like those phospholipid bilayers, and the matrix that we were looking at. So we also have this series of membrane-bound proteins, and these membrane-bound proteins, some of them are channel proteins. And notice they're carrying the hydrogen ions, so they're really those transport proteins we had talked about. So they're going to carry hydrogen ions against the concentration gradient. So up here in the intermembrane space, we have lots and lots of hydrogen ions. So this is the high concentration. And down here is the low concentration of hydrogen ions. So the natural process, or well, both are natural, but the process that doesn't require energy would be for the hydrogen ions just to reach equilibrium and not flow. But we don't have that, or we can't have that for this. So here we're going to start with the 2-NADH are going to come into this electron transport chain, and they're going to drop off those high energy electrons, and that's what you see here. So this little E negative is a high energy electron, and that high energy electron, those high energy electrons are going to pass through the electron transport chain, giving the electron transport chain energy to do their work. This allows the hydrogen ions that are down here, and remember, notice that the 2-NADH leave is 2-NAD positive. So that means not only did the high energy electrons get dropped off, but also the hydrogen ions. And those hydrogen ions are going to pass through those transmembrane proteins against the concentration gradient using the energy from those electrons. So the hydrogen ions are going from an area of low concentration to an area of high concentration. So that's why we need that energy, because this is active transport. Okay. Once this whole area up here fills up with hydrogen ions, they're going to want to diffuse back into the matrix. And they do that diffusion by or through the ATP synthase. And since it's an enzyme, or since it has A's in it, it's an enzyme. An ATP synthase is an enzyme that also has a protein channel through it. And the hydrogen ions can diffuse back through ATP synthase. So here are the hydrogen ions diffusing back through ATP synthase. When they diffuse back through ATP synthase, that causes ATP synthase to spin. And that spinning mechanical force helps to, or it doesn't help to, it adds energy to ADP, and that's stored in that third phosphate bond. So the movement of hydrogen ions diffusing back through ATP synthase causes it to spin. The spinning force is the source of energy that ADP picks up, stores in that third phosphate bond to become ATP. So that's where this half-charged molecule here picks up its energy by the spinning and becomes a fully charged molecule, ATP. And what we have left over is some of those used up electrons. And those used up electrons get picked up by water and by oxygen and hydrogen ions to become water. And this is where that final waste product, the water, is produced during cellular respiration. Okay, so in the end, what we have is, this is kind of a breakdown of what we could get from glucose. So during from glucose during cellular respiration. So during glycolysis, we make 4 ATP, but we use 2. So our net gain is 2 ATP. Okay. Also during glycolysis, we make 2 NADHs. This is for 1 molecule of glucose. Those 2 NADHs go to electron transport chain, but the ones from glycolysis only make 4 ATP. Okay, so 2 each. The other thing that's produced during glycolysis is pyruvate. Pyruvate goes into the acetyl-CoA reaction, where it produces, each pyruvate produces 1 NADH, but we have 2 pyruvates that were made during glycolysis. So the acetyl-CoA reaction produces 2 NADH. But notice that the NADHs produced during the acetyl-CoA reaction produce 3 ATP each, because we made 2 pyruvate, or made 2 NADHs from that 1 molecule of glucose, and they each made 3. So that's different than what we had here with the NADHs produced during glycolysis. So one of the questions on your worksheet is going to be to figure out why that is, or to at least hypothesize why that is. Okay, the Krebs cycle makes 2 ATP directly, but the Krebs cycle for 1 molecule of glucose, you get 6 NADHs. Each of those NADHs makes 3 ATP to give you a total of 18. And then the Krebs cycle also makes 2 FADH2, but those FADH2 only make 2 ATP each. So one of the other questions on the worksheet is, why does FADH2 only make 2 ATP in the electron transport chain, But each of the NADHs made in the Krebs cycle makes 3. So as a hint, I would tell you to look at the diagram of the electron transport chain. So the total amount of ATP that is our net gain from 1 molecule of glucose, the theoretical yield would be 36 ATP.